I want you to imagine a microscopic assassin. But, like, this isn't your typical killer.
Right. It doesn't operate the way you'd expect.
Exactly. It doesn't even need to enter your house to do its deadly work. It doesn't, break a window or pick a lock.
No forced entry at all.
Yeah, it just sets up camp on your front porch, makes itself totally comfortable, and then quietly slides the perfect, untraceable weapon right under your front door.
And, I mean, that defining trait, that total lack of physical invasion, that front porch strategy fundamentally rewrote the rules of what early scientists believed an infectious disease even was.
Which brings us to today. Welcome to today's deep dive. We're looking at a huge stack of microbiology textbooks, historical medical records, and modern epidemiological data.
We really went through it all for this one.
We did. And we're using all of that to investigate Corynebacterium diphtheria, which is the bacterium behind diphtheria.
A truly fascinating organism.
It really is. And our mission today is to figure out not just what this bug is, but exactly how it works. Because, honestly, this Victorian-era nightmare completely flips the script on what a pathogen actually is.
It really does. It challenges all our basic assumptions.
Right. So the entire story we are going to unpack today revolves around this central theme, the illusion of the pathogen. We're going to see how a hijacked microbe, a terrifyingly efficient molecular machine, and a disease that operates entirely by remote control just, changes everything we know.
And I should add, this isn't just some abstract history lesson. The mechanics of how this organism operates challenge how we define bacterial infections right now today.
Yeah, this isn't just about the 1800s.
Exactly. The real story here is about a weaponized biology that still demands, well, constant vigilance from global health systems.
So true. Okay, let's unpack this.
Yeah.
Before we look at this secret weapon being slid under the door, we have to look at the assassin itself, right?
You have to start the source.
Yeah. If you go back to the 1880s, early scientists were starting to realize this wasn't a typical microscopic invader. I mean, most bacteria, when we think of them, act like a home invasion.
Right. They kick the door down.
Exactly. They break into your bloodstream, they multiply, they spread through your deep tissues, and they just physically wreck the place. But Corynebacterium diphtheria doesn't do any of that.
No, it doesn't. To understand its strategy, we really have to look at its biology. This is a gram-positive, club-shaped bacterium.
Club-shaped?
Yeah. Actually, the genus name, Corynebacterium, it comes from the Greek word korine, which literally translates to club.
Oh, wow. Okay, so when you say gram-positive in this context, what does that practically mean for the bacteria? Like, for its survival?
Well, it means the bacterium has a very thick, robust outer cell wall.
Okay.
That thick wall allows it to hold on to specific laboratory stains, which is how we identify it in the lab. But practically speaking, it makes the organism really tough.
Like it has armor.
Sort of, yeah. It can survive outside the body on surfaces for a bit, but once it finds a host, once it gets to you, it thrives in oxygen, attaching itself to the mucosal surfaces of your throat, or your tonsils, or, sometimes broken skin.
And that is where it stays, right?
That is exactly where it stays. It's non-modal.
Meaning it can't swim.
Right. It can't swim around, and it does not typically invade the bloodstream at all. It just sits on that mucosal porch, as you called it, as a local colonizer.
It's just hanging out. And the sources say the organism was first spotted by a German-Swiss pathologist named Edwin Klebs in 1883.
Yes, Klebs.
He was looking at stained smears from the throats of diphtheria patients and saw these weird club-shaped bacteria clustered together. But seeing them in a sick patient and proving they actually cause the disease are two very different things.
They are completely different things. The rigorous proof came a year later, in 1884, from Friedrich Loeffler.
Oh, Koch's assistant, right?
Yeah, exactly. A former assistant to the famous Robert Koch. Loeffler managed to culture the organism using a blood serum medium.
Which was a big deal.
A massive deal. It's a technique known as Loeffler's serum slant, and, amazingly, microbiology labs still use it today.
That's crazy that it's still used.
It works perfectly. So by isolating it, growing it in pure culture, and reproducing a diphtheria-like illness in guinea pigs, he formally satisfied Koch's postulates.
Right, and Koch's postulates being that famous gold standard checklist scientists use to prove a specific bug causes a specific disease.
That's the one.
You find the bug in the sick animal, you grow it in a lab, infect a healthy animal, and see if it gets the same disease.
That is the checklist, yeah. And for a long time after Loeffler completed it, the bacteria was actually known as the Klebs-Loeffler bacillus.
Catchy.
Right. But Loeffler ran into a massive paradox during his experiments.
The paradox being where the bacteria was actually found.
Exactly. When he autopsied the guinea pigs that had died from the disease, the physical bacteria were still just sitting exactly where he put them at the site of inoculation in the throat or on the skin.
Okay, wait. So if the bacteria is just sitting on the porch, why are the animals dying? Because the autopsies showed massive fatal damage to distant organs, right? Specifically the heart and the nerve.
They did. And Loeffler guessed the bacteria must be secreting some kind of soluble poison that traveled through the body, but he couldn't isolate it.
He just didn't have the tech?
Right. The real breakthrough happened in Paris in 1888. This was Émile Roux and Alexandre Yersin at the Pasteur Institute.
Okay, what did they do?
They took a broth culture where the bacteria had been growing for a while, and they poured it through an unglazed porcelain filter.
And that type of filter is, like, incredibly fine, right? Like a microscopic coffee filter. So the physical bacterial cells simply cannot pass through.
They get completely trapped.
But any dissolved liquids or chemicals can just flow right through.
Exactly. So they took that sterile, completely bacteria-free liquid, the filtrate, and injected just that liquid into healthy animals.
No bacteria at all.
None. And the animals developed the exact same full-blown disease. Paralysis, heart damage, death.
Wow. So they proved that a secreted chemical, a toxin, was doing all the killing.
Yes. It was the first rigorous proof in history that a microbial disease could be caused by a chemical weapon rather than the physical presence of the bug itself.
Which is just a massive paradigm shift in medicine. I mean, scientists later used that conceptual template to understand things like tetanus, botulism, and cholera.
The proximity of the bacteria literally doesn't matter. The reach of the toxin does.
But that brings up a huge question. If this toxin is just floating freely in the blood, why doesn't it destroy every single cell it touches? Like, why does the throat bacteria end up destroying the heart and the nerves specifically?
That comes down to the actual architecture of the diphtheria toxin itself. It belongs to a category called AB toxins.
AB toxins.
Yeah, you can visualize it as a two-part molecular machine. The B stands for binding and the A stands for active.
Okay.
The B subunit's entire job is to float through the bloodstream, completely ignoring everything until it finds a very specific receptor on the surface of human cells.
So if we're using our analogy, the B subunit is essentially a lockpick looking for a very specific keyhole.
That's a great way to look at it. And that specific keyhole is a receptor called heparin-binding EGF-like growth factor.
HBEGF, right?
HBEGF, exactly. And those specific receptors happen to be highly, highly abundant on heart muscle cells and on the Schwann cells that protect your peripheral nerves.
So that's why. The geography of the disease's damage is dictated entirely by the toxin's chemical preference for those specific receptors, not by where the bacteria lives.
Precisely. The bacteria is in the throat, but the lockpick only fits the doors on the heart and the nerves.
That is wild. Okay, so once that B subunit lockpick finds the receptor on a heart cell and attaches, what happens next?
Well, the human cell is basically tricked into swallowing the toxin whole.
Just gulps it down.
Pretty much. The cell membrane bubbles inward and engulfs it in a process called endocytosis. Once the toxin is inside, the environment becomes acidic, which allows the A fragment, that's the active payload, to break away and escape into the cytoplasm.
And the cytoplasm is like the jelly-like main body of the cell where all the cellular machinery operates, right?
Right, it's the factory floor of the cell.
And this is where the biological devastation happens. The sources say the A fragment uses a chemical process called ADP ribosylation to permanently disable a unique amino acid called diphthemide.
ADP ribosylation. It sounds incredibly dense.
It really does.
But mechanically, it's quite simple. The A fragment basically glues a bulky chemical tag onto a specific cellular protein, and the protein it targets is called eukaryotic elongation factor 2, or EEF2.
Okay, and why does the cell care about EEF2?
Because your cells absolutely need EEF2 to build new proteins. Without it, protein synthesis completely starves.
So if EF2 is what the cell uses to build proteins, and this toxin glue a bulky tag onto it, it's jamming the gears.
It's completely jamming them.
So what does this all mean? Instead of the toxin blowing up the cell like a bomb, this is more like sneaking into a massive car factory. Yeah. The assassin sneaks onto the main assembly line and steals one highly specific, irreplaceable wrench.
I love that analogy.
Right. And without that one specific wrench, the entire assembly line just grinds to a halt. No cars get built. The factory dies.
The cell literally starves itself of proteins and undergoes cell death. But what makes this biological sabotage truly terrifying is its catalytic efficiency.
What do you mean?
Well, the A fragment isn't just a bullet that gets used up once it hits a target. It's an enzyme.
Oh, no.
Yeah, it can systematically move through the cytoplasm, gluing that bulky tag onto thousands of EFU molecules one after another after another.
Wait, really? So a single microscopic lockpick gets in, and it just wanders around the factory stealing wrenches forever?
Basically, yes. Toxicologists absolutely revere the sheer potency of this mechanism. It is theoretically possible for a single molecule of the air fragment, just one reaching the cytoplasm, to shut down protein synthesis so completely that it kills the entire cell on its own.
One molecule.
One molecule. It is a masterclass in targeted biological warfare.
Okay, but to build a molecular weapon of that scale, you need a serious operation. If we go back to the battlefield, the throat, where the bacteria is actually sitting, we find out that the bacteria isn't just quietly resting on the porch while the weapon does the work. There is a brutal local battle happening too, right?
Oh, absolutely. The toxin destroys the local epithelial cells in your airway just as efficiently as it destroys heart cells.
So it's doing local damage too.
Yes, and as that local tissue dies, it becomes necrotic. The body's immune system rushes in to fight it, causing massive inflammation and leaking fibrin, which is the body's natural clotting mesh.
Right, trying to patch things up.
Exactly. But all of this dead tissue, this fibrin, red blood cells, and the bacteria itself, they all fuse together into what is called a pseudomembrane.
Yeah, the sources describe it as this tough, gray-white, leathery layer. Actually, the Greek root diphthera means leather or hide.
That's exactly where the name comes from.
And this leathery membrane is terrifying because it can grow and spread down into the larynx and trachea, physically suffocating the victim.
It's horrific. Historically, that mechanical obstruction was one of the two main ways diphtheria killed, right alongside the toxin-induced heart failure that would strike a week or two later.
It's just a nightmare on two fronts.
Yeah.
But here's where it gets really interesting. The massive plot twist in modern microbiology, and this goes back to our theme of the illusion of the pathogen, is that Corynebacterium diphtheriae is not the true architect of this disease.
It really isn't.
The bacteria is entirely incapable of manufacturing the diphtheria toxin on its own.
Which is mind-blowing.
Hold on. If the bacteria can't cause the disease on its own, why do we even call it a bacterial infection? Isn't it technically a viral disease?
Well, in a functional sense, you could argue yes. The only way this bacteria can produce the weapon is if it has been infected itself by a specific virus.
A virus for bacteria?
Yes, a bacteriophage called Corynebacterium beta. This virus carries the tox gene, which is the actual genetic blueprint for the toxin.
The bacteria catches a virus, and that virus turns it into an involuntary weapons factory. It's an innocent bystander.
Essentially, yes. The biological term for this is lysogenic conversion. The virus integrates its own DNA directly into the bacteria's genome. And suddenly, this relatively harmless throat microbe starts pumping out one of the most lethal toxins known to biology.
Incredible.
If a strain of C. diphtheriae doesn't have this specific phage, it's called non-toxigenic. It might cause a minor sore throat, but it cannot cause diphtheria.
So we've been blaming the wrong culprit all along. The bacteria is just the delivery vehicle for a viral blueprint. And understanding that completely shattered some massive medical debates, didn't it?
Oh, it completely upended the field. Early clinicians used to argue intensely over different strains, or biovars, of the bacteria.
Right, the older doctors?
Yeah, they noticed that the gravis biovar seemed to cause severe deadly disease, while the mitis biovar was usually much milder.
Gravis meaning grave, mitis meaning mild.
Exactly. But modern genomic science revealed that was an illusion. The severity isn't about the bacterial strain at all. It is entirely dependent on whether or not the bacteria is carrying that specific bacteriophage.
And the sources point out that this viral phage can infect other closely related bacteria too, right? Like Corynebacterium ulcerans, which is typically found in cattle or pets.
Yes, it can jump.
So someone could get classic human diphtheria from a farm animal just because the virus jumped to a different bacterial host.
They absolutely could. Clinical microbiologists had to completely redefine the disease. Diphtheria isn't strictly about one specific species of bacteria anymore. It's a functional category.
Wow.
The disease is caused by any Corynebacteria that happens to have been hijacked by this toxin-producing virus.
So humanity is facing this brutal, highly efficient, remote control weapon that isn't even built by the pathogen we thought it was. Before modern antibiotics, How on earth did we fight back against this?
Well, the fight back led to one of the greatest triumphs in medical history,
What?
which unfortunately quickly gave way to one of its absolute darkest tragedies.
Okay, let's start with the triumph.
In 1890, Emil von Behring, working in Berlin, discovered that you could immunize animals against the toxin. Their immune systems would create antibodies that neutralize the poison.
Right, he harvested their blood serum and used that antitoxin to cure animals that were already infected. The antibodies would just flood the bloodstream and neutralize the toxin before it could lock onto those heart and nerve cells.
Exactly, and this led to the famous Christmas Eve miracle of 1891.
Miracle.
Yeah, von Behring was in a Berlin clinic, and he used this experimental animal serum to treat a dying child. And it worked beautifully. It was hailed as the first successful use of serum therapy in human medicine.
And he won a Nobel Prize for that.
He did. He won the very first Nobel Prize in medicine in 1901 for the discovery.
Amazing.
Yeah.
But to produce this life-saving antitoxin at a commercial scale, they had to scale up the animal part. They relied on hyperimmunizing horses, right?
Yes. Horses?
They would inject a horse with harmless amounts of toxin, let the horse build up massive amounts of antibodies, and then harvest large batches of his blood serum.
Horses were ideal because they are large animals that can produce a significant volume of serum.
Makes sense.
But that manufacturing system had a catastrophic blind spot regarding quality control, and that blind spot was exposed in St. Louis in 1901.
The St. Louis disaster. What exactly went wrong with the horse serum?
Well, the city of St. Louis kept a retired milk wagon horse named Jim to produce their local supply of diphtheria antitoxin. And Jim had reliably produced over 30 quarts of life-saving serum.
Good boy, Jim.
But Jim contracted tetanus.
Oh, no.
The final batch of serum collected from him right before he was put down was contaminated with live tetanus bacteria and its own lethal toxin.
And because there were no rigorous safety or purity checks required by law at the time, no one tested the final batch.
No one tested it. The contaminated antitoxin was bottled, distributed, and administered to children suffering from diphtheria.
That is just...
At least a dozen children died agonizing deaths from tetanus. They were given a medicine meant to save them, and it killed them.
It's completely heartbreaking. A life-saving therapy became a vehicle for death simply through poor manufacturing and literally zero oversight.
It was a massive scandal.
But the outrage over those children dying actually changed the regulatory landscape of the entire country, didn't it?
If we connect this to the bigger picture, the public horror over the St. Louis disaster was the direct catalyst for the U.S. Biologics Control Act of 1902.
Which was the first law of its kind.
Yes. This was the very first federal law in America regulating the safety, purity, and potency of vaccines and serums. It mandated laboratory inspections and licensing for manufacturers.
Wow.
Modern medical safety regulations were quite literally written in the blood of those children in St. Louis. It proved that understanding the science of the antitoxin wasn't enough. We needed robust, regulated systems to protect public health.
We regulated the treatment, which is huge, but antitoxin is reactive, right?
Okay.
You only give it once someone is already sick. To actually defeat the disease, we needed a way to prevent it entirely. Which brings us to the modern era. And the final illusion of the pathogen, the illusion of our own victory.
This is a really crucial point. In the 1920s, a scientist named Gaston Ramon figured out how to treat the diphtheria toxin with heat and formaldehyde to create a toxoid vaccine.
Okay, wait. How does formaldehyde turn a lethal molecular weapon into a vaccine?
The formaldehyde essentially bakes the toxin. It changes the physical shape of that lethal A fragment so it can no longer destroy your cellular machinery.
So it breaks the active part.
Right, but it leaves the B lockpick structure perfectly intact. Your immune system can still take a mugshot of the B subunit and learn to recognize it, but the weapon itself is deactivated.
That is incredibly elegant. It's safe. It teaches your body to neutralize the toxin, and routine childhood immunization practically wiped the disease off the map in wealthy countries.
It did.
I mean, we don't see children suffocating from leathery membranes in the throat anymore, so the general public just assumes the disease is cured.
And that victory is an illusion due to a fundamental misunderstanding of what the toxoid vaccine actually does.
Because it doesn't kill the bacteria.
Exactly. The vaccine only neutralizes the chemical toxin floating in your bloodstream. It does absolutely nothing to stop the actual Corynebacterium from colonizing your throat, surviving on your mucosal tissues, or being spread to other people.
So if I'm fully vaccinated and perfectly healthy, I could literally be carrying the bacteria on my porch right now.
You absolutely could.
It's not a microscopic loaded gun waiting to go off. It's more like weeds lying dormant in a garden. They're totally harmless to me because I have the vaccine protecting my cells, but they are just waiting to spread to someone whose garden isn't protected.
That is a much more accurate way to look at it. The vaccine protects you from the toxic effects of the disease, but it doesn't clear the organism from the human population. Non-toxigenic strains and even toxigenic strains circulate constantly.
Which is terrifying.
Furthermore, the immunity provided by the toxoid vaccine wanes over time. It is not a lifelong shield. Adults require a TD or Tdap booster roughly every 10 years to keep those antibody levels high, and it is highly recommended during every pregnancy to pass that protection to the newborn.
But when people assume the disease is gone, they stop getting boosted. The sources point out some famous catastrophic failures that happened because of that exact assumption, most notably in the former Soviet Union in the 1990s.
Yes. During the Soviet era, rigorous centralized vaccination programs had practically eliminated diphtheria. But when the USSR collapsed, the public health infrastructure, supply chains, and routine childhood vaccinations collapsed right along with it.
So the dormant weeds were still circulating, just waiting for the population's immunity to drop.
Exactly. When that herd immunity fell, diphtheria exploded across Russia and neighboring states. We're talking over 100,000 cases and several thousand deaths in just a few years during the 1990s.
Over 100,000 cases of a Victorian disease.
It took massive international emergency mass immunization campaigns to finally bring it back under control by the end of the decade.
And it's not just a story from the 90s, is it? We've seen massive outbreaks recently in Yemen, in Venezuela, and in the Rohingya refugee camps in Bangladesh. Basically, anytime war, economic collapse, or a humanitarian crisis disrupts routine healthcare, diphtheria comes roaring back almost instantly.
This raises an important question about how we view modern public health. Public health agencies actually use diphtheria as a sentinel indicator.
The sentinel indicator.
Yeah. If you want to know if a region's health system is starting to crack, you watch for diphtheria outbreaks. Because the pathogen is always circulating in the background, the moment vaccination rates dip, this disease slips right back through the front door.
It's like a stress test for a society's healthcare.
Exactly. Diphtheria control is not a one-time historical achievement. It is a permanent maintenance condition.
It is a constant battle of vigilance. So for you listening, let's quickly recap this incredible journey through the illusion of the pathogen. We started with a club-shaped bacteria just hanging out on the porch.
Harmless on its own.
Right. We discovered it's actually an innocent bystander that gets hijacked by a virus, turning it into a factory for a molecular lockpick that targets your heart and nerves. We saw how the attempt to cure it drove the creation of our first medical safety laws, and how the vaccine we rely on today doesn't actually kill the bug, it just disarms the weapon.
It is a profound story of biology, human history, and the necessity of public health infrastructure. But I'll leave you with this final, rather chilling thought to ponder.
Oh boy.
If a completely harmless throat bacterium can be transformed into a mass killer simply by catching a virus that hands it the blueprints for a molecular weapon, how many other harmless microbes living inside your body right now are just one viral infection away from becoming the next major pathogen?
Wow. It really makes you think twice about the microbes sharing your porch. And seriously, take this as your reminder to go check your medical records and make sure you've had your Tdap booster in the last 10 years. Thanks for joining us on this deep dive.